Yes, so very good evening, my dear students. Today I am going to start with this animal kingdom second part. So part 2 we are going to start. So the topic is going to be last day what we have completed. What we have completed, Gracie, if you remember. We have classified animal kingdom on the basis of 10 different points, I think. Can you just remember what are the points we have talked about? The first point, let's start with that. Level of organization is the first point for Ashika. Level of organization is the first point. What is the second point? On the basis of level of organizations, there are 4 different types: cellular, tissue, organ, and organ system. Second is what? The second is on the basis of their symmetry. On the basis of symmetry. Arijit, on the basis of their symmetry. When we talk about the symmetry, there are 3 different types: asymmetrical, bilateral symmetrical, and radial symmetrical. Radially symmetrical. Just check it out if the voice is over. Okay. Now the third type, and one we can add another one called biradial. In biradial there is bilateral and radial, both of them together. Today we are going to talk about one of them. What is the third way that we can classify them? The third way is on the basis of their body cavity, called coelom. There can be 3 different types: acoelomate, pseudocoelomate, and eucoelomate. So this is number 3. Number 4 is on the basis of their germinal layers. So we have classified them into 2 types. Number 1, diploblastic and triploblastic. Diploblastic is having ectoderm and endoderm, and triploblastic is having 3, ecto, meso, endoderm. Fine. So 4th is over. 5th, on the basis of their digestion, we can classify them into 2 types. One is intracellular digestion, and the one is extracellular digestion. Next, 5th is over. 6th, the 6th is what? On the basis of their digestive system, we can classify them into 2 different types: complete and incomplete. When it is complete, when they have got anus and mouth at 2 terminal ends, we call it complete. Incomplete means anus is not present. Next question, sir. Is there any organism having no anus? We are going to talk about that. Yes, there are. So 6th way is over. 7th way, on the basis of their being able to cope up with the present environment. There are 2 different types: homeothermous animal, poikilothermous animal. Homeothermous animal means what? They are warm-blooded animals, those who can maintain their body temperature with that of the environment. Gracie, tell me about yourself. You are warm-blooded or cold-blooded animal? Warm-blooded. Cold-blooded animals are called poikilothermous animals, like those organisms who cannot regulate their body temperature with that of the environment, like frog, like snakes, reptiles. So we are going to talk about that. This is the 7th one. 8th way that we can classify them is on the basis of the body plan. Have we talked about that? Body plan. So there are 3 different types: cell aggregate type, blind sac body plan, tube within a tube body plan. Come on. The 9th one, on the basis of their fertilization, there can be 2 types: external fertilization, internal fertilization. On the basis of their development of mouth and anus in their embryonic stage, we can classify them into protostomia and deuterostomia. I think I have talked about that. Yes or no? No? Protostomia, deuterostomia? Okay, we are going to talk about that then. And tell me some more. Is anything is there? Yes. On the basis of their segmentation. We can classify them into metamerism segmentation and pseudo-metamerism. Metamerism means what? In every other segments, some of the organs are present in repeated fashion. And in case of pseudo-metamerism, they appear to be having many segments, but they don't have repeated organs in every segments. What is the example of pseudo-metamerism? The example of pseudo-metamerism is tapeworm or Tinea solium. Clear? We are going to talk about that also, no issues. Have we talked about this? I think we have talked about segmentations. Okay. So these are the ways that we can classify the different organisms. So 11 or 12 points are there. Fine. On the basis of the development, there can be of two different types. Direct, indirect. Direct means having no larval stage. Indirect means having larval stage. So majority of the invertebrates they show indirect development. Is it clear? Okay. Let us start with, we are going to start with the phylum today. We are going to start with the animal kingdom, especially which part? The phylum, invertebrates phylum. We will start with phylum Porifera. But before we get into phylum Porifera, let me discuss about protostomia and deuterostomia. It's very, very important. What is it? On the basis of their embryonic development, we can classify them into two types. We can classify them into two types. And what is that? Okay. On the basis of, so we can classify animals. We can classify animals on the basis of their development, embryonic development. Embryonic development into two. Embryonic development into two. One is called protostomia. Protostomia. Proto means what? First. Where from we have named the other term by starting with proto? Protozoa. First animal. Zoa means animal. Protozoa. So protostomia, where from the term stomia has come? From stomology. Study of mouth is called stomology. So first mouth develops, then anus. Organism where first mouth develops and then anus. Example, all invertebrates, which starts from, come on, phylum Porifera, then Nidaria, Ctenophora, Platyhelminthes, Ashelminthes, Annelida, Arthropoda, Mollusca, Echinodermata and Hemichordata. All of them they have got protostomia. No exception is there. From phylum Porifera to phylum Mollusca, all of them they have protostomia. From Echinodermata to all chordates, they have got this. What is it? Deuterostomia. Deuterostomia. What is deutero? Later. Later what? What later? Mouth later. So first what?
Anus.
First anus development and then mouth. What is the best example of it? The best example is phylum Echinodermata, which is an invertebrate. It starts from phylum Echinodermata till chordata, till chordates. All of us, we have got deuterostomia. So what about human being? In the embryonic stage, what is that structure which first originates? Anus or mouth? Anus first, then mouth. Clear? So we are all deuterostomes. Is this clear? Any issues with this? Come on, Yogesh. So let's start with the topic called, this is how we can also classify organisms. So let us start with the first phylum, and the phylum is phylum Porifera. Porifera. Now, break this term into two. What is that? Pori plus fera. Fera means bearing. Fera means bearing or having. Pori means pores. So basically they are pores bearing organisms. They are pore bearing organisms. Clear? So their body is having billions of pores. Their body is having billions of pores. If this is the body, say for example, this is another one, fine, and they are sessile structure. They have got mouth-like structure. This is a bigger structure called mouth. And they have got billions of pores all around, minute pores all around their body. Clear? They have minute pores all around their body. These minute pores are called ostia. These minute pores are called ostia. And this ostia is having the pore called inhalant pore. What is the inhalant means? As because this poriferans which are completely aquatic, they are exclusively aquatic. Exclusively they are aquatic. So you cannot get any one of the examples which are land dwellers. So they are completely aquatic, number one. So aquatic means they can remain in marine as well as they can remain in fresh water. So very, very important, you have to know the examples of marine and fresh water. Okay. Some of them are okay, freshwater sponge. Okay, now marine and freshwater sponge. Okay, marine and freshwater. Now, so completely they are aquatic. That means they are going to be present at the base of the water bodies, at the surface, and they are going to remain stuck with some of the point. And if an organism is present stuck with some structures or substratum, we call that organism as sessile or sedentary. Sessile or sedentary. So they are sessile in nature. What is the meaning of sessile? They are attached to the substratum. Attached to the substratum. Understand? These are the terms that you have to use. Attached to the substratum. Clear? Now. So water gets inside through ostia, inhalant, and goes out through the large structure mouth called osculum. It is a large pore called osculum. It's a large pore called osculum. So osculum is exhalant in nature. Exhalant. So what is exhalant? The water moves out, exits. Clear? So water gets inside through ostia, goes out through osculum. So definitely this is a special characteristics in them. It's a special characteristics in them. Now make a wild guess. I'm using two systems. One is water canal system and the one is water vascular system. And give a wild guess which we are going to call about poriferans. Water canal or water vascular. It's a water canal system. So this particular characteristics that water gets in through the ostia, goes out through osculum. This is what is very, very important and it's unique with all poriferans, and they have got water canal system. They have got water canal system. It's a special feature of them. Okay. Let us talk about porifera. When we talk about porifera, what is the study of porifera called? The study of porifera is called as poripherology. It is called parazoology. What is it called? Parazoology. Parazoology. Okay. Now, the structure of this poriferans. Achha, what is this example? The example is about cycon, which is also called as skypha. Just have a look in your book. Skypha. Skypha. Cycon or skypha is the example of it. Now, if you see this structure, let me get into the next page. If you see the structure, okay, if you see the structure of this poriferans or skypha or cycon, you can see that this poriferans the common name of these poriferans are called sponges. They are called as
Sponges.
So from sponges, you can get one term which is called as spongin fibers. So the body is having spongin fibers. The body is having spongin fibers, which makes it flexible. Secondly, a little bit of rigidness is there due to the deposition of silicon and calcium. Due to the deposition of silicon and calcium. So we call these structures as siliceous and calcareous spicules. Siliceous and calcareous spicules. Siliceous and calcareous spicules. So if they have got siliceous and calcareous spicules, it gives them a rigid structure. But a little bit of what? Flexibility is there also, and that is due to the presence of spongin fiber. Presence of spongin fiber. Is this clear? Any issues with this? Yogesh? Got the point? That means when we are going to classify them into different classes, we are going to see that on the basis of presence of calcium, silicon or spongin fiber, we are going to classify them into 3 categories. Clear? Chalo. Now, what is special about them is they show what kind of level of organization. What kind of level of organization they show? They show cellular level of organization. Cellular level of organization is shown by them. Clear? Though they are multicellular, but still they show cellular, very, very ancient type called they have got cellular level of organizations, though they are multicellular. That means cell to cell interaction is not there. Clear? Cellular level of organizations. Think about its coelom. Do they have any coelom? They have got a body cavity. The cavity that you can see inside, there is a hollow. And this hollow structure is called as spongocele. It's called as, the hollow structure is called as spongocele, which is also called as paragastric cavity. Also called as paragastric cavity. Any issues with this? Also called as paragastric cavity. Clear? Now, they don't have any coelom. We call them as acoelomate. We call them as acoelomate organism. Acoelomate organism. Okay? Acoelomate organism. Is this clear? Now, what kind of symmetry they show? They are asymmetrical. You cannot divide their body into 2 equal halves in any plane. But some of them they can show what? Bilateral or radial symmetry. Basically they are asymmetrical, but some of them they can show bilateral or radial symmetry. Now, they are acoelomate. What kind of germinal layer they have got? They are diploblastic in nature. What is diploblastic? How many layers they have? 2 germinal layers. What are they? Ectoderm and endoderm. Ectoderm and endoderm. So they are diploblastic in nature. What kind of fertilization they show? External or internal? They show external fertilization. They show external fertilization. They show external fertilization. Okay. When we talk about this water canal system, water gets inside through ostia, goes out through osculum. Yes? Internal fertilization. Yes, internal fertilization is there, but mind you, what kind of, when we talk about the reproduction, internal fertilization is predominant. Internal fertilization is predominant, but some of the advanced type, they go for external fertilization. So this is dominant. Internal fertilization is dominant. If you understand why we are talking about this external fertilization, then we have to talk about that the reproduction process is by asexual means, and also it can take place by sexual means. By sexual means, they can they can go for gamete formation, and by the help of the gamete formation, they can go for sexual reproduction. But when we talk about asexual reproduction, if you have your class 12 book, old book, in page number 7, what you can see is a structure was given, which now has been omitted. No, sure. Yes. This is a structure which is called as gemmules. Fine. This is gemmules. And these gemmules, this structure called gemmules, fine, is there in the old book of class 12. It's not there in the new book. This chapter has been omitted. Now, these gemmules are the structure which are asexual structure in sponges. So the asexual structure is gemmules. And you know, in asexual structure, asexual way of reproduction, there is no variation. But in sexual, there is a variation is possible by the help of gametes. So basically, they show internal, but some of them, which are advanced type of poriferans, they show external fertilization. Clear? When we talk about water canal system, the water canal system, it is a process by which they can go for 3 particular type of organ system. They can help in respiration, digestive system, because I will talk about that, how they help in digestion. And the last one is excretory system. So by the help of water canal system, they can go for these 3 processes: respiration, digestion, and excretion. Let me talk about the digestion. See, if we see this particular structure, this is what? Ostia, opening. Water, when gets inside, but with this water, the food can also get inside. Encircling this ostia, there are some cells, special cells, which helps in what? There are some special cells which helps in trapping the food. And these special cells which helps in trapping the food are called choanocyte cells. They are called choanocyte cell. And these choanocyte cells, which are also called as collar cells. So by the help of these collar cells, they can trap the food present inside water. And if you just see the outer structure, so this is the layer, the outer layer of the poriferans. The outer layer is made up of pinacoderm, and the inner layer consists of a germinal layer, a dermal layer called choanoderm. Fine? The collar cells are present over here with some cilia-like structures. So pinacoderm, at the peripheral part of the pinacoderm, they have got these kind of cells called as choanocyte cell or collar cells, which helps in trapping the food. Is this clear? Yes. Very good. Now. What kind of development they show? Direct or indirect? They show indirect development. And in this indirect development, they have got 2 types of larva. The larva is amphiblastula larva, and the one is called parenchyma. This is another larva. Parenchyma is solid larva, and amphiblastula is hollow larva. That means if larva is present, what kind of development you can expect them to have? Indirect development. Any issues? So larva is there. Yes. What else we can talk about them? We can classify phylum Porifera into 3 different classes. The first one is called as calcarea. The second one is called hexactinellida. Hexactinellida. And the last one as demospongia. Demospongia. So tell me, Arijit, if you talk about calcarea, what you can get from the name itself? They have got more of calcium depositions. They have got more of calcium depositions on their body. So calcium deposition is there. Calcareous spicules. Calcareous spicules is predominant. Calcareous spicules is predominant in them. Fine. So what should be the example of this? The example of calcarea is Sycon, which is also called as Skypha. Fine. Sycon is the best example of it. When we talk about hexactinellida, hexa means what? 6 or 7? 6. So they have got 6 rows. 1, 2, 3, 4, 5, 6. 4, 5, 6. 6 rows of this kind of structure. 6 rows of what? They have got siliceous spicules. 6 rows of siliceous spicules. That means they have got silicon as deposition, but they are present in 6 rows. The best example, mind you, the example is very important. The example is Euplectella. The example is Euplectella, which is called as a Venus flower basket. Venus flytrap is insectivorous plant. Venus flower basket. And this Euplectella, which is a sponge having hollow structure inside, spongocoel inside, and this particular Euplectella inside it, what you can see is some of the shrimps are present. And the name of the shrimp is called Spongicola. The name of the shrimp is called as Spongicola, and they keep their tentacles outside this hollow structure. So any predator which predates on Euplectella fears out of seeing these tentacles. What has come out from that particular hollow structure? So who gets survival edge due to this? What? This combination of 2 organisms. Euplectella gets the survival edge. So for Euplectella, it is positive association. And for shrimp, they get shelter, but come on, that is 0 for them. What is 0? What is this kind of association where species A and species B is present? One is getting benefited, other one is neither being benefited nor being harmed. Commensalism. Commensalism. So this is the best example of commensalism. Commensalism. I think in class 12, some of the page I have talked about, page 232 of old book. 232 of old book, class 12. Or new book. 171. Huh? Saprophytes. No. In mango, the best example is mango, and mango is having orchid. Some of the orchid plant, they get stuck with mango tree. They never draw nourishment from the mango tree. It's not a parasitic plant. So for mango it is 0, neither being benefited nor being harmed by this association. But orchid gets what? Shelter. So for orchid it is positive, for mango it is 0. Have you got the page? Page 171? No. It's a new book. Page 232 for old book. And 200, it is there in page 197. Commensalism. See? Positive and 0. Page 197 of new book. See, the commensalism is positive and 0. Gracie, why don't you come over here and see? Have a look. Commensalism. 197. Class 12.
Species B যদি benefit 0 species B যদি benefit 0 হয় তাহলে থাকে কেন?
না, actually হামও করছে না, কোনো লাভও হচ্ছে না, but হামও হচ্ছে না. It's a mutual relationship. We call it as population interaction. So two of the species, they are interacting, where it's not like always they should have a mutual beneficial role. Clear? So if there is a mutual beneficial role, we could have called it as mutualism or symbiosis. So it's a perfect example of commensalism. Now mind you one thing about Euplectella, they are called as famous wedding gift. Famous wedding gift. Where? In Japan. So they are normally found in close to Japan Bay of Japan. And it's a famous wedding gift. Why famous wedding gift? It depicts till death do us apart. It depicts what? Matte dam tak saath nibhayenge. Till death do us apart. So it's very, very important for your NEET examination. Fine. Euplectella is having 6 rows of siliceous spicules, and that's why they come under Hexactinellida class, and they are the famous wedding gift in Japan. Clear? What is even more important about them? That they show commensalism with some of the shrimps, and the name of the shrimp is Spongicola. Any issues with this? What is the next example under this Hexactinellida? Example is glass rope sponge. Find out, is there, can you find this in NCERT? Glass rope sponge or Hyalonema. Glass rope sponge is the next example of glass rope sponge. Is the next example called as, this is the common name of Hyalonema. They are also having 6 rows of siliceous spicules. They have got 6 rows of siliceous spicules. Clear? Okay. One of the examples we have talked about over here, Calcarea is Sycon. You can again, another one, it can be Leucosolenia. Leucosolenia is one of the example of Calcarea. So we have got 2 examples of Hexactinellida. What is that? The first one is Euplectella. The second one is glass rope sponge called Hyalonema. Called Hyalonema. Any issues? What is the last one? Demospongia. What is the last one? Demospongia. Arijit, tell me, what is so very important about this term? What you can find it out from this term? Demospongia. What is there inside this term? Spongia. That means what they can have more of? Spongin fiber. They have more of spongin fiber. Demospongia. So they have more of spongin fiber. What is the example? What is the example of Demospongia? Class Demospongia. In your book, find out. Have they given you this term called Euspongia? Can you see this example? Euspongia. It is called as bath sponge. The common name is bath sponge. So they are very much what? They are very much soft due to the presence of spongin fiber. So they have got less of what never makes them tough. Siliceous and calcareous spicules are less in them. Vaishali, are you getting me? So they are also called as bath sponge. What is the next one example being given in your book? Spongilla is a freshwater sponge. Freshwater sponge. I told you earlier also that majority of them are marine, but few of them are freshwater. So freshwater, example is Spongilla. You see the diagram in your book. It's very, very important in NCERT. In NEET, what they can do is, they can just give you the diagram and tell you to find out what is it. Spongilla has been given, I think. Yes? So, next. What else we can have as an example?
This is the eye.
Yeah.
This is the eye.
Eye? No. It's not eye. They have got billions of pores. Where from you are getting eyes?
Spongilla.
No. They're not eyes. Fine? Okay. Now, some of the examples like, yes, Euspongia, Spongilla. These are the examples of Demospongia. So, Demospongia means what? They have got more of spongin fiber. More of spongin fiber. So, that's all about kingdom Porifera. That's all about kingdom Porifera. Just go through your NCERT line by line. You will be getting everything clear. Clear? Yes? Let's get into the phylum Nidaria. Nidaria. Phylum Nidaria. Next is phylum Nidaria. Nidaria. Just break the term. Nid plus area. Area means area. Area means bearing, having. Bearing or having. मेरे पास है. क्या है मेरे पास? मेरे पास nid है. What is nid? Nid stands for nidoblast cell. Nidoblast cell. What is this cell? These cells are nothing but they are stinging cells. They are stinging cells. They are present where? In the tentacles. So the best example of this is Hydra. So they have got tentacles. They have got tentacles and they have got a mouth. This mouth is called hypostome. This mouth is called? Stome means what? Mouth. Hypo means what? In the lesser part. So if this is present as a sessile hydra, it can move by somersault. The moment it makes a somersault, the tentacles are going to remain at this, and this is the structure. So the mouth is present at hypo, called hypostome. Clear? Yes, somersault. So they can move like this. These are tentacles. These are tentacles. In their tentacles, they have got stinging cells, and these stinging cells are very, very unique, and that's why they are called Nidaria. Got the idea? What is nid? It has come from nidoblast cells called stinging cells, and they have got neurotoxins. Yeah, sometimes they can sting on you, like some of the jellyfish, which comes under Nidaria. If they sting on your thighs, then there will be a numbness in your thigh. Fine? So they are going to sting you. Sting, they have got stinging cells. The stinging cells are also called as nematocysts or nematocyte or nematocytes. Clear? Now, this is about Hydra. Yes?
Stingray?
No, stingray is a fish. It's a condyctis. It's a cartilaginous fish. In Bengal, it is called as Shankar Mach. Yes, yes. Now, they have got a structure which is hollow. See? Hollow. Yes? Tell me what kind of body plan they have. Single mouth, no anus, called blind sac body plan. So they have got blind sac body plan. So we have missed one point in phylum Porifera. What is the body plan? Cell aggregate type of body plan is there in phylum Porifera. So if they have got blind sac body plan, that means over there in sponges, they have got cell aggregate type. They have got cell aggregate type body plan. Is this clear? Because they show cellular level of organizations. They don't have interaction between cells, though they are multicellular. So what kind of aggregation, what kind of body plan they can show? Cell aggregate type. So they have cell aggregate type of body plan. Any issues? Great. Fine. Okay. So Nidaria gets into this. Get into this. What kind of body plan? They have got blind sac body plan. So if you consider the cavity, Nidaria, they are also called as coelenterata. Coelenterata. Again, you break this term into three. Seal has come from Koilos. Fine. Seal has come from koilos. Entera has come from enteron. And ata means bearing. Ata means bearing. So they have got koilos bearing. What is enteron? It has come from enterology. Tell me, if you have got some problem in your stomach or intestine, whom you are going to show? Which doctor you are going to reach? Cardiologist or gastroenterologist? Gastro means stomach, entero means intestine. So enteron means intestine. So enterology means what? Intestine. So they have got a hollow type of intestine structure. Seal means cavity. Seal means cavity. So cylindra, that's why they are also called as coelenterata. And the cavity is called coelenteron. The cavity is called? The cavity, these are tentacles, the cavity is called? Coelenteron. So the cavity is called coelenteron. Okay? Seal means cavity, enteron means intestine. Okay. Now, this particular cavity, we can also call it as gastrovascular cavity. Very, very important. Gastrovascular cavity. Yogesh, tell me, what is the cavity present inside poriferans? Inside poriferans, what is the cavity called? Spongocoel. Spongocoel. C-O-E-L. Spongocoel. The cavity present inside sponges are called spongocoel. Clear? So, this is done. Next, what kind of level of organizations they show? Level of organization. Very good. For the first time, they show tissue level of organization. For the first time, which shows for the first time tissue level is cnidarians. Clear? Next. What is their symmetry? What is the symmetry? Majority of them are radial symmetry. They show radial symmetry. Radial symmetry. What about their coelom, body cavity? Acoelomate. Acoelomate. They don't show any coelom. What is the germinal layer? The germinal layer is diploblastic. Diploblastic. Okay, body plan we have already talked about. What is the body plan? Blind sac body plan. Blind sac body plan. Tell me some more information. Give me some more information about it. What kind of fertilization? What kind of digestion? Intracellular or extracellular? Okay, we have missed on the point. In porifera. In porifera. What kind of digestion is shown by them? Intracellular digestion. What kind of digestion is shown by poriferans? Intracellular. That means they have got cellular level of organizations. Can you expect tissue level? Can you expect any different type extracellular? Cell ke bahar digestion hoga? Never ever. So they have intracellular. When we talk about cnidarians, the digestion is intra as well as extra. Intracellular and extracellular both. So they show intracellular as well as extracellular digestion. Clear? Tell me about their development, direct or indirect? Direct or indirect? They show indirect development because they have got larva, and the name of the larva is planula and ephyra. Ephyra. Planula and ephyra. These are the name of the larva. These are the name of the larva. So they show indirect development. Yes. Yes. Now we are getting into the most important thing, and that is their main feature. What is the main feature? They show Polymorphism. What is the meaning of the term poly? See if tell me. Poly means what? Poly means many. Poly means many. Morph means what? Form. So they are having many forms. They can remain in many forms. What are the two forms that they are having? The main forms that they can have or they are having is polyp, and another form is medusa. One form is polyp, another form is medusa. When we talk about polyp is think about hydra. So they have got tentacles like this. This is hydra. Fine. So think about this is a stalk-like structure and this is an umbrella-like structure. Just to make umbrella, this is an umbrella-like structure. Clear? Erect umbrella. Make it upside down. These are tentacles and this is a jellyfish. The stalk has turned up. So the jellyfish having tentacles. You have seen jellyfish like this only? Yes? Whatever picture that you have got about jellyfish. Jellyfish scientific name is Aurelia. Aurelia is a jellyfish. The common name is jellyfish, scientific name is Aurelia. Clear? So medusa is this shape. But they can remain in polyp as well as in medusa form. And they can interchange their form. And that's why we call this interchanging of this form as alternation of generation. We call this alternation of generation. That means polyp can change into medusa by PAM. Polyp can change into medusa asexually. And medusa can change into polyp sexually. And this changeover is also called as alternation of generation, or we can also call it as metagenesis. So star mark point about all Nidarians that they can show this metagenesis. Very, very important. Metagenesis. So polymorphism is the most important term about phylum Nidaria. Special feature. Polymorphism, alternation of generation, or we can call it as metagenesis. Metagenesis. Clear? Now, let me tell you about the different class under phylum Nidaria. So there are three different classes. The first class is about class Hydrozoa. The next one is Scyphozoa. And the third one is Anthozoa. In Hydrozoa, what is unique? In the class Hydrozoa, what is unique? The uniqueness is about they have got both polyp and medusa and they can interchange among themselves. So both of them are dominant. The best example of this is Hydra. And also Obelia. I think you have seen Obelia is there in your book called as Seafur. Obelia. So Hydrozoa, the examples are Hydra, Obelia, Seafur. So they can both polyp and medusa are dominant over here. In Scyphozoa, the dominant is what? The dominant is medusa. Medusa is dominant and polyp is reduced. Polyp is reduced and medusa is dominant. Tell me the example of it. Definitely Aurelia. Come on. Aurelia is the best example of this. When talk about Anthozoa, they have got polyp as the main dominant one and medusa is reduced. Example of Anthozoa. Tell me the example of Anthozoa. The first one which comes in your mind immediately is this structure. What is the structure? This is sea anemone. They have got tentacles like this. This is sea anemone. And this sea anemone, the scientific name of this sea anemone is Adamsia. Is there in your book? And mind you, sea anemone, they have got small tentacles. And they show biradial symmetry. They show? So if they have tentacles in the upper portion, definitely the polyp structure is dominant, just like umbrella or hydra. Clear? Now, what is so very special about sea anemone? They also show commensalism with hermit crab. They also show commensalism relationship with hermit crab. They sit at the back of hermit crab because they are slow movers. In some of the marine aquarium, you can see them. If some of them are having marine aquarium, the very common thing that they keep along with some marine fish is sea anemone. They're slow movers. Sometimes the sea anemone can sit at the back of hermit crab. It's a crab. It's a crab, hermit crab. And they sit at the back of the hermit crab. Who is faster? Hermit crab is faster. So sea anemone gets a free ride. But what about how hermit crab gets benefited by the association of sea anemone at the back? Sea anemone, when it sits at the back, it secretes some chemical which is having obnoxious smell. And the predator of hermit crab, when it tries to feed on hermit crab, due to that obnoxious smell, they don't feed on them. A very bad smell. So the predator, they are not going to feed on this kind of hermit crab having sea anemone on their back. Clear? So who gets survival edge? Hermit crab gets survival edge. So positive about hermit crab and zero for sea anemone. This kind of positive and zero relationship is called commensalism. So very, very important. You have to remember, whenever we talk about sea anemone, what should immediately come in your mind? Hermit crab. What should immediately come in your mind? Hermit crab. Now this is sea anemone. The other term for the sea anemone is metridium. And the example of the sea anemone is metridium. It is also a sea anemone. So in your book, sea anemone, Adamsia is being given. See, Adamsia is being given in your book. What else? Some of this anthozoa, they have got calcium carbonate depositions. Some of this anthozoa, they have got calcium carbonate depositions. Due to this calcium carbonate depositions, they are called as corals. They are called as corals. So there are some corals called brain coral. And the scientific name of this brain coral is Meandrina. The scientific name of this brain coral is Meandrina. Clear? Very good. So this is brain coral. Some of the corals, they look like mushroom. So the example is about mushroom coral, and mushroom coral, the scientific name is Fungia. The scientific name is Fungia. Is it clear? Now, mushroom coral is Fungia. Mushroom is Fungia. Mushroom, you know already, fungi. Next is what? Next is, see some of the examples being given as sea fan, Gorgonia, 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 also called as sea fan, Gorgonia. See, there is an example called Pennatula, Pennatula, sea pen. So they look like pen only. What is this? At the top. And they look like sea pen, called Pennatula. Sea pen. Fan and modun, sea fan. So Gorgonia, they are flat shaped structure and they look like fan only. And fan ta bhalo kore ghore na, ghor kore ghole, jera Gorgonia. Okay? Very good. So this is Gorgonia, Pennatula, sea anemone. Okay, we are missing on a point about hydrozoa, and that is Physalia. See in your book, Portuguese man of war. Portuguese man of war. They actually look like that. They actually look like that on Portuguese man of war. They look like warrior. Have a look. Have they given any diagram? No. Okay. Okay. Portuguese man of war. Physalia. Fine. They have got gas glands. They have got gas glands, and they are poisonous glands. The poisonous glands that they have. Physalia. Very, very important. So these are the examples. These are the examples. See if we have missed any of the examples which has been given in your book. I don't think so. Me and Rina, we have done it. Yes. Obelia, we have done it already. Aurelia, free swimming. Yes. Larva we have talked about. Fine. Shia is there. So that's all about phylum. Nidaria. That's all about phylum. Nidaria or Coelenterata. Coelenterata. Let's get into the next one. What is the next phylum? The next phylum is about Ctenophora. Phylum Ctenophora. The third one is phylum Ctenophora. Fine? Ctenophora. Now, what is so very important about phylum Ctenophora? Phylum Ctenophora, they are basically called as, the common name of this is C wallet is okay, but they are called comb jellies. They are called as comb jellies or sea gooseberries. Sea gooseberries. Fine, or comb jellies or sea walnut. So they are common names. Now, Ctenophora was initially under phylum Nidaria. It was placed initially under Nidaria. What is the reason that they have been taken out of this Nidaria? Is they have got tentacles, but without nidoblast cells, without stinging cells. So Nidaria, they don't have stingings. No stinging cells are present in Ctenophora. No stinging cells are present in Ctenophora. So you can see in the figure, they have 8 rows of comb plates. 8 rows of comb plates. 8 rows of comb plates. Fine, they have got some tentacles. By the help of these tentacles, they can help in what? By the help of these 8 rows of comb plates, what they can do is they can locomote. So there is a locomotory organ. They can move from one place to the other. Now, 8 rows of comb plates. What is so very special about Ctenophora is Ctenophora, they show bioluminescence. They show bioluminescence. That means they emit light. They emit light. Bioluminescence. Clear?
Because of chemicals.
Phosphorus, again, we have talked about this. Phosphorin, in the earlier classes, we have talked about. Okay? Yes. Now, phylum Ctenophora, tell me what kind of symmetry, what kind of level of organizations you can expect them to have. What kind of level of organizations? Yogesh, tell me. Nidarians, they were having tissue level. इसके पास भी क्या है? Tissue level of organizations. Clear? What about their coelom? What about their coelom? Acoelomate. Okay. Tell me the symmetry. It's very, very important by seeing the diagram. Seeing the diagram, you can see these are the. This is the comb jelly-like structure. Symmetry, radial. Radially symmetrical. Fine. They are radially symmetrical. Tell me what kind of acha, they have got their mouth, and they don't have excretory system. They don't have excretory system. It's very important. Clear? Okay, we have missed one thing in Hydra. In Nidarians, they have got nervous system and they have got nerve nets. They have got nerve nets. In Hydra, They have got nerve nets, and that's why tentacles are very, very sensitive. These tentacles, they have got nerve nets. So what is the nervous system they have? A very, very primitive type, and they have got nerve nets. Okay? When we talk about ctenophora, they have got no excretory system. Okay? Nerve nets, tentacles are very, very sensitive. The moment they come in contact with any other organism, they will sting on them. So they're highly what? Sensory in nature. So they have got nerve nets. By the help of nerve nets, they can, they can sting on them. Yeah. So that is it. Yes, under phylum Ctenophora, in your book you can see what kind of digestion they show, both intracellular and extracellular digestion. Fine? Both intracellular and extracellular digestion. So what kind of digestion they show? Again, digestion shown by them is both intracellular and extracellular digestion. Okay? What about in case of Cnidarians? They have got same intracellular and extracellular. What in case of phylum Porifera? Only intracellular. Okay. Ctenophora, they have got two classes. We can classify them into two classes. One is tentaculata, and the one is nuda. Tentaculata. So these are two classes. There are two classes of phylum Ctenophora. Phylum Ctenophora. They have two classes. One is tentaculata. Example is pleurobrachia. Pleurobrachia. Example is pleurobrachia. The example of class tentaculata. Pleurobrachia. An example is Tenoplana. Tenoplana under tentaculata. What about nuda? In tentaculata, what is there? A special point is there about tentaculata. What is that? They have got tentacles. Ata means what? Bearing. Ata means bearing. Bearing what? Tentacles. Bearing what? Tentacles. Bearing what? Tentacles. Nuda, they have no tentacles. Very good. No tentacles. Now, under example of nuda, what is the best example? The best example is Surjo Bero. তাড়াতাড়ি কেউ বলে এরকম? I said Bero. No, Bero example. বারোই দিলি তুমি, হ্যাঁ? তো বুঝবে কি করে? আর একটু সিরিয়াস থাকতাম তারপরে তো হতো ব্যাপারটা। So Bero is the example. Got the idea? Gracie, what is Bero in Bengali? Chale jao. To ghabra gaya. Okay. What is that? So nuda is Bero. B-E-R-O-E. Bero. Huh? Okay. So this is it. Clear? So I think only one example is given in your book. একটা example দিয়েছি দেখ. Only one example has been given in your book. Only Tenoplana, I think. No, pleurobrachia. Pleurobrachia has been given. Tenoplana. Oh, done. So we have got another one. Okay, the fertilization is external and with indirect development. The fertilization is external. The fertilization is external. So think about it. Inside the body, the fertilization never takes place. It takes place outside the body. So it is an external fertilization present in them. So that's all about phylum Ctenophora. So initially it was present inside phylum Cnidaria. What is the reason for that? What is the reason that they have been separated? Lack of stinging cells. They have tentacles, but they don't have tentacles. They don't have stinging cells. Let us get into phylum, and the phylum is phylum Platyhelminthes. Phylum. Next phylum is phylum Platyhelminthes. Now, let me give you some idea about this. Platyhelminthes. Tell me, what is the meaning of the term platy? Platy means flat. Platy means flat. So basically they are flat. What is helminth? The study of worm is called helminthology. The study of worm is called helminthology. So study of worm is called helminthology. So what is worm? Helminth means what? Worm. So they are flat worm. So basically what kind of flat are they? They are dorso-ventrally flattened. Say for example, this is tortoise. What is it? Tortoise. Now, this is a shell that they have. The shell is called carapace. We call it dorsal carapace. So what is the meaning of the term dorsal? Upper part of the body is called dorsal. Against gravity is called dorsal part. It's called?
Dorsal.
Towards gravity, they have got this flat portion, and this flat portion is called plastron. So they have got ventral plastron. So what is the meaning of ventral? Towards gravity. So if we call it that they are fine, this tortoise, when the result has been declared and father of tortoise is beating on this one, what is going to happen to the tortoise? Dorso-ventrally flattened? Dorso-ventrally flattened? समझ में आया कि नहीं आया? Dorso-ventrally flattened. So that is what kind of flat are they? They are dorso-ventrally flattened. Is this clear? So now their body has just become like ribbon. Dorso-ventrally flattened. Clear? What is the best? What is the utility of this? The best example of this phylum Platyhelminthes is tapeworm. And say 30-40 feet long tapeworm can remain in a small space in your intestine by folding themselves, by keeping them folded. Think about it. This is dorso-vent. That means majority of this phylum Platyhelminthes, they are parasites. They are
Parasites.
But if I talk about some exceptions, so exceptions becomes very, very important. Some of them are a non-parasitic. And the example that you already know is about planaria. Is about?
Planaria.
You have seen this figure? Yes, planaria. If you cut the planaria into 3 pieces, this piece is going to make the whole body. This piece is going to make the upper 2 bodies, and this one is going to make the whole body. Planaria is a non-parasitic Platyhelminthes. Clear? So the best example of regeneration. So this is planaria, which is the best example of regeneration. Any issues with this? Come on. So what kind of flattened? Dorso-ventrally flattened body. Okay. Dorso-ventrally flattened body. So whenever we talk about this Platyhelminthes, what comes in immediately in our mind is about the structure. For the first time, you can see an organism having head-like structure, and the formation of head-like structure is called as cephalization. Called as cephalization. So what is the meaning of the term cephalization? Formation of head. Till now, we have not seen a single organism having head-like structure. For the first time, we are finding over here, this is a head-like structure. So the term cephalic means head. Cephalic means? So formation of head is there for the first time in this tapeworm. So you can see in the head-like structure, they have got suckers. And along with the suckers, they have got hooks by which they can grab hold of the intestine. So 1, 2, 3, 4 and the back 5. The whole of the head, they have got suckers and hooks. They have got suckers. They have got suckers and hooks attached on them. Hooks attached on them by which they firmly attach with the intestinal wall to take what? To take all the nourishment that you are having in your body. Clear? So this is the head-like portion in a tapeworm. And their body is like this. Their body is like this. They have got many of the segments. Many of the segments. So this head-like structure is called as scolex. It's called as scolex. And they have got many small initial part of the head portion. Near to this head portion, all these segments are smaller. And these are called immature. These are called as immature proglottids. Immature proglottids. But the moment you go a little bit at the next portion of the body is having more of bigger segments, and these are called as gravid proglottids. They are matured one, gravid proglottids. In some gravid proglottids, they have got testis and ovary. Say this is testis and this is ovary. This is male part and this is female part. That means their body is monoecious or dioecious.
Monoecious.
We call it as hermaphrodite. A single body having both the sex is called hermaphrodite. So basically they are hermaphrodites. Bisexual body. Clear? Same body having testis and ovary. Vaishali, tell me, is testis and ovaries present in every other segment? No. Testis and ovary are present only in mature segments called mature proglottids. Yes? Every other gravid proglottids is having testis and ovary. But what about this? Immature proglottids, they don't have testis and ovary. So can we call them as metameric segmentation? What is metameric segmentation? Every other segment contains in earthworm, every other segments of earthworm, it contained what? Nephridia. The excretory organ are present in repeated fashion in every other segments. What about this? No. So we call this as pseudo-metamerism. Pseudo means false. It seems to be having metameric segmentation. Actually it is not. Or pseudo means false. So they show pseudo-metamerism. So this is the structure of tapeworm. The scientific name of tapeworm is Tinea solium. Tinea solium is the scientific name. Tinea solium is the scientific name of it. Any issues? Now what they can do is, see this tapeworm, which is a pork tapeworm. This is called as pork tapeworm. What is pork? Pig. Fine. So there's another one called beef tapeworm, called Tinea saginata. Beef tapeworm. Tinea. The beef tapeworm is Tinea saginata. Tinea saginata is the beef tapeworm. Okay. Now, what is so very important about tapeworm? They can by themselves, they can cut off their body part. Just like you have seen in lizard, they can take off their tail portion. Fine. Just like that, they can shed off this later part of the body, and the process by which they can do it is called apolysis. Apolysis. So they can shed off their body parts. Think about it. The person who is having this, tapeworm, where? In the intestine. The adult tapeworm, they are going to throw their body part. Which body part? The body part which contains testis and ovary. So in this testis and ovary, there should be definitely eggs present. When they are going to let it off, this portion is going to be present within the stool or feces of this particular person. this person has gone in the open to defecate. In the open fields, this person has gone for defecation. Within this particular, within the fecus or feces or stool, what is present? Eggs of tapeworm. Who is going to feed on this? Who are those coprophagous animal which feeds on the human stool? Pig. So one of the pig followed that person and fed on that particular stool. Eggs are going to go inside the pig's body. So think about it. Eggs will be consumed by the pig along with the stool. Those eggs are going to go to the stomach first. And there's a larva which is called hexacanth larva. And then it is going to go into the bloodstream. Heart is going to push the particular larva to every part of the body. And say, for example, a person, the pig is having those larvae in the meat, in the flesh. So when a person goes over there to buy the pork meat, a pig meat, that particular pig meat or flesh contains the eggs of or the larva of the tapeworm. This particular larva is called CBSE larva. The larva is called CBSE larva. And the meat having this CBSE larva is called as measly pork. It's called as?
Measly pork.
Do you think all the pig meat contains this larva? No. That means how many hosts? So the moment a human feeds on this CBSE larva, the larva gets inside the human body and inside intestine it is going to grow into adult. Again, the life cycle is going to go on. Again, that adult is going to remove off their gravid parotid part, which will contain the eggs, and eventually again the pig or some different pig is going to feed on that. So how many hosts are required? How many body are required for the completion of the life cycle of tapeworm? 2. One is pig, another one is human. Pig and human. So how many hosts?
2.
Tell me which one is primary host and which one is secondary host. Wild guess. And what is the meaning of primary host? What is the meaning of primary host? The host body where the sexual reproduction takes place is the primary host. So sexual reproduction takes place in human body. Primary host. Secondary host is where the asexual. Okay, that's all for now. Clear? Next day we are going to continue with this phylum Platyhelminthes. We are going to have a continuation class about this one. Sure, that's it. Okay? Any issues? Sure.